I'm Agnieszka Niemark, and this is the Boyce-Thompson Institute Centennial Oral History Project. It's September 30th, 2023. My interviewee today is Professor Robert Last. Robert joined the BTI's Plant Molecular Biology program in 1989 and worked at the Institute until the late 1990s. He was also an adjunct professor in the Department of Genetics and Development at Cornell University. We are recording this interview at the Boyce-Thompson Institute in Ithaca, upstate New York. Robert, first of all, thank you so much for accepting our invitation to join the project.
It's my pleasure, thank you.
Before we talk about your affiliation with BTI and the early years of the molecular biology program at BTI, I would like you to start by telling us a little bit about your educational background and maybe about your interest in studying chemistry, who inspired you to study chemistry and later molecular biology.
Yeah. So before graduating high school, I went to the New School for Social Research, which in 1975, 76. And at that point, the New School in New York City, in Lower Manhattan, really only had one undergraduate program, which was a one-year program for students to go for their first year in college instead of their last year in high school. And this was a really important time for me, I now realize intellectually, because the New School, I studied a fair bit of science and math there, but there was no such thing as a course that was only about biology or only about calculus or what have you. The faculty really took a very, to use almost a trite term, but it was true in this case, holistic approach to teaching. And so, for example, in biology, I took a year of biology with Paul Mankiewicz. Paul was a graduate student studying, of all things, plant cell wall proteins, but his real interests were in the principles that govern structure and function of plants. And the cell wall was, for him, a conduit into this interest. And so we studied Darcy Thompson. We studied, actually, Richard Dawkins, The Selfish Gene, the year it came out. We just got a lot of background and a real emphasis on the integration of biology and physical sciences and so forth. And at the time, of course, I had no idea that how important this would be for the future, but it was really formative. Then really was not sure that I wanted to go to college per se. The New School, it was really a very unusual program, and it was not a program that really kind of dropped you on your head and made you think about things differently. But I did go to Ohio Wesleyan University for a year. I went for 4 years, but the first year thinking, well, it's either this or Culinary Institute. You know, if this formal education doesn't really work, I'd like to perhaps become a chef. I got absolutely hooked. You could say addicted to chemistry and biology through some wonderful teachers and mentors, the first of whom hooked me through general chemistry, a fellow named Harold Wilcox, who at that time was already quite a distinguished senior faculty member, who I believe he failed me on my first midterm exam. We had midterms every 2 weeks. And that could have ended up being quite depressing. But when I went to see him, he said, well, you know, how much did you study? And you seem very interested in this stuff. And I said, well, you know, I told him. He said, well, that's about a quarter of what you need, the work that you need to put in to really learn this material and so forth. And he went on to become quite inspirational to me. At the same time, Ohio Wesleyan had a very strong biology, and still does. It was almost 100% organismal biology. Molecular biology was just in its infancy and certainly not dominant at Ohio Wesleyan. So I really got this wonderful education in organismal biology, taking courses in mycology, fungal science, ornithology, birds, from a wonderful mentor, Jed Burt, who passed away in the last decade or so, unfortunately. We were freshmen together, I think. We both started at Ohio Wesleyan at the same time. He as an assistant professor, and learned a lot of biology. So chemistry and biology. Well, it's not obviously biochemistry, but somehow this led to interests in biological chemistry as time went on.
How did you make your choice about doctoral studies?
Oh, choice. You make it sound like one really chooses, or at least if you're me, you really choose. I think it sort of, in some ways, it's luck. Many ways, it's fortune, luck, and in other ways, it chooses you. What is it? The wand chooses the wizard in Harry Potter parlance. So I really, really wanted to study the evolution of protein evolution. Turned out that, and protein structure, function, and protein evolution, from a biochemistry course that I took from a man named Danny Vaught, who again started teaching about the same time that I started college, and I TA'd for him the first time he taught biochemistry at Ohio Wesleyan. It turns out that because of the way that genes were cloned and proteins were sequenced in the 1970s, really only highly conserved proteins, I think that might be even almost 100% accurate, were ones that we had DNA sequences for, and so on and so forth. So it was the wrong time for a topic which I now study, and for the last 15, 20 years, my lab has been able to study because of technologies changing. So I went to Carnegie Mellon University to not a new department, but a reconceived biological sciences department. There were a number of very influential senior folks there. Elizabeth Jones, somebody who had a huge influence on me. Her rigor, her intellect, and her personality were really influential. But Beth and others had hired a number of young faculty who were practicing what was, I guess, becoming known as molecular genetics, sort of the convergence of classical genetics and molecular biology. And I went to Carnegie Mellon to study protein structure using NMR, realized that I did not have the mathematical tools, the physical chemistry background to do that, and ended up becoming a geneticist in the laboratory of John Wolford. Again, a relatively, I guess I was his second graduate student, a recent hire, someone who cloned some eukaryotic genes the hard way, really hard way, back when you had to work really hard to find the sequence of a single gene, let alone, in his case, 5 or 10 ribosomal protein genes. And so the transition just happened naturally, it was natural and coincidental, and that chance is really luck and chance are really important, I think, in career development.
So we are talking here, Robert, about early molecular genetics, right? And we are now in the early 1980s.
Yes.
How you decided about, continuing in particular field and choosing then postdoctoral studies?
Oh boy, here we go with luck and chance again. In this case, in around 84, I started applying for postdoctoral training. And I really wanted, so I'd been using yeast, baker's yeast, Saccharomyces cerevisiae, as a model eukaryotic organism, and got to ride this amazing wave of technology and community development in yeast. Well, I really became very interested in cell biology and applied to laboratories that were pioneering the use of genetics to study cell biological processes. Well, I arrived in the office of Gerald Fink, who at the time was a relatively early in his time at Whitehead Institute. He had been at Cornell here in I believe in genetics and development department for the first part of his professional career. So there's some Cornell history there. And Jerry said, you know, if you like, you can do that. We have some projects. But I just got back from California and was talking to Ron Davis, a really influential scientist still, I believe, at Stanford University, Elliot Meyerowitz at Caltech, and maybe others. And there's this plant called Arabidopsis that you know they've convinced me is going to be the next big thing in plant genetics and plant biology. And he sent me off to think about it. And I did, and spoke to some people I know who knew something about plant biology, and was convinced that this was a really great idea.
So can you explain a little bit more, Robert, how this plant became a model plant for biology studies, right, or cell studies?
Yes, small genome, which became important in the genome era. Very fast generation time, so that if you're doing genetics, you don't have to wait 6 months to harvest seed. You wait 6 weeks. So for example, in Jerry's lab and then in my lab at Boyce Thompson Institute, we were able to, I'm not good with math, but you know, maybe at least 12, or 8 to 10 cycles of growth per year. And you could do experiments, in rapid succession. It's not the 20 minutes for cell division of E. coli or hour or 3 hours of Saccharomyces, but you know, quite good. Also, and small. So one in a growth chamber of the sort that we had at MIT, which was really just kind of put together with not quite gum and baling wire, but you know, we conceived in a small space you could grow a lot of plants and do a lot of genetics and then eventually a lot of genomics.
So could you just explain, Rob? So basically, you applied this earlier knowledge that you gained from studying yeast, or bacteria, fungi into plant, right?
That's right.
Yeah, that's the early plant studies.
Yeah, that was really fortunate in the sense that Jerry was trained and was and is still, he's retired now, but a consummate geneticist, trained as a microbial geneticist, and so. What you know, Jerry immediately went to the concepts, the approaches that he had learned and that I had learned, for that matter, in microbial genetics, and applied some of those, the ones that we could, to a multicellular, a complex development organism. In this case, a plant. And so, yeah, we grew plants on Petri plates. We subjected them to, you know, the seeds of Arabidopsis are tiny, 50,000 in a little gram of seeds. So we could grow them on Petri plates with inhibitors. We could do all sorts. We could grow them so that they would grow on vertically, so that we could observe the roots as they grew down the agar, and just all these different tricks. Small is beautiful in this case. Yeah.
Okay, so you were at the Whitehead Institute for Biomedical Research around 1986-89, and around the same time at BTI in 1988, they decided to establish a first molecular biology program at the Institute. That was during the time of Ralph Hardy as president of BTI. Could you explain in what circumstance did you learn about BTI, or what was the connection from the Whitehead Institute to BTI?
Again, chance and wonderful people played a role. In this case, in the guise, not in the person of Andre Jagendorf, who was a very distinguished plant biochemist here at Cornell. Andre joined Jerry's lab within a month of my joining. I think I was there in May. I started in May of 86, and Andre maybe in June or July as a visiting scientist. We were all crammed. There were like 10 of us eventually in this one small laboratory in Jerry's lab, really built more for 4 or 5 people. So there was all this great interaction. And Andre and I really hit it off. I knew nothing about plants. Agnes, I knew absolutely nothing about plants. And Andre knew huge amounts. His career, unfortunately, he's now deceased, but Andre had a storied career in plant physiology and plant biochemistry. And so he taught me a lot about plants. And then when he came back at the end of his year, I believe it was a year in Jerry's lab, he came back to Cornell, learned about this burgeoning or planned set of hires in plant molecular biology. I think maybe Steve Howell had already been hired as director of the program. And I think he sent me, or as we used to do, probably put in the mail with a stamp on it, a copy of the ad for the position that David and I ended up not competing with each other, but getting hired for, right? Yeah.
So then your interest in molecular plant biology was growing at the time, and you thought that might be a good idea, right?
That's right. Well, I mean, Cornell is certainly a well-known, well-established still, and then place to study plants. And Jerry certainly encouraged it because of his experience here, and interviewed, was offered the position. Met up with Ralph Hardy. Maybe I'm sure I met Ralph during the interview, but I remember quite well meeting up with Ralph. He was in Cambridge, Mass, for a business meeting, and we had breakfast together at his hotel in Harvard Square. Remember this very well. There are some moments in one's life that one remembers quite well, and we had a really great conversation about his vision and the Institute's vision for continuing to grow and expand in molecular biology across the Institute.
So eventually you joined BTI in 1989, and during the presidency of Ralph Hardy, there was a lot of reforms happening at BTI, that's between years 85-95. Could you tell us, Robert, how the Institute looked like? Can you talk a little bit about the other programs at the Institute that were here when you arrived to BTI, and how molecular biology was slowly integrated into these other programs and also established as a single program?
Yeah, so a bit of that was through hiring. Gary Blizzard certainly was hired into the plant protection program. Gary with a very strong set of skills in baculovirus biology. It's always dangerous to name names because I could be leaving someone out, and I apologize if I am. But and then I guess the plant molecular biology program, the three of us, Steve, David, and I, started establishing our own research programs. And collaborating with folks at BTI, at Cornell. And so I'll just give an example of a very, very important, for me, a very important infiltration, if you will. It was really a mutual infiltration. So I had been working on amino acid biosynthesis since joining Jerry's lab, and I was quite interested in metabolism. I this background in biochemistry, in chemistry and biochemistry, in genetics, And I realized that, first of all, that there were some amazing facilities here at BTI that had been created through the plant, the environmental biology program. Some really great expertise, some great facilities, some great minds, and I realized that there was I don't know if I realized, but it turns out that there was a really good synergy, set of possibilities between some of the organismal physiologists, folks who are interested in what happens to a tree when it's out in acid rain or in a sulfur dioxide-rich environment. These were from the unfortunate dirty coal-burning electric power plants, which now that we worry about them because of carbon dioxide. Back then, acid rain and sulfur dioxide and ozone effects on plants. But my interests were in these little tiny plants, not in great big plants, but they're really great synergy. So, for example, Bob Amundsen, who was a staff scientist, I guess, or a faculty member at the time in environmental biology, and his group with Rich Reba, and my group, Jiayang Li, and originally Jiayang Li and others in my group, established a collaboration really quite informally, which led to some really, some of my most highly cited papers, perhaps the first times that genetic mutants had been used to study environmental stress adaptation or prevention mechanisms, in this case UVB, which was a big concern because of stratospheric ozone depletion. The stuff that was coming out of people's refrigerators, out of air conditioning units, which was going up to the stratosphere and causing depletion of the ozone layer. And by the way, that was originally, as I understand it, discovered by someone who had been an undergraduate at Ohio Wesleyan completely. Anyway, that's a completely different story, a Nobel Prize, et cetera. But so, yeah.
And you mentioned to me, Robert, that thanks to great technicians working at BTI, you were able to make some facilities, some lab equipment that help you to expose plants, right, to the large amounts, or how you describe, of the UV
That's
light,
right.
or intense UV light.
That's right. There had been a really an amazing, almost like a torture chamber, this amazing growth chamber that had been designed with very tight UV control and the ability to ramp up. So plants growing outside, experience dawn all the way through high noon to dusk, right? Well, usually we just turn the lights on in the morning, we turn them off. They had designed a chamber to try to simulate outdoor environment. They had all these great chambers that were good for doing air pollution kind of biology, generating ozone, generating sulfur dioxide, exposing them to plants.
We discussed some of these technological, advances with people from the environmental biology previously. So yeah, it was done here.
Yes. So we benefited greatly from their expertise. The facilities that they had been using for trees, for soybean, if I remember correctly, for other crop plants, and their expertise. Bob Amundsen was a really great collaborator. We came from completely different, he was an ecophysiologist, I was a molecular geneticist, and for a while we had a really great collaboration, yeah.
What were the main findings of this particular research?
Right, so the work focused on the UV work, focused on the question of, plants sit outside, they get bombarded by UV all the time. As long as there's sunshine, there's ultraviolet of different amounts and so forth. So why are plants able to survive? That was the fundamental question. We might develop pigmentation in our skin. What do plants do? And there were all sorts of theories in the literature, some of them half-baked, some of them supported by science, about UV sunscreens that plants make constitutively all the time, and that are induced when you take a plant from no or low UV to high UV. I thought some of these compounds made sense as sunscreens, but having been trained as a chemist, I thought the others really were not UVB absorbing compounds. So there were mutants available in the public domain at that point that were deficient in these different sunscreens at different steps in the pathways. And so we used those mutants to ask the very simple question, well, if molecule X is an important UV protectant, then genetically preventing the synthesis of molecule X should make the plants quite sensitive. And in some cases, the answer was yes. And in other cases, the answer was not so much. And so we were able to publish that work. And for me, it was the first step toward using genetics as a way of understanding how plants cope with environmental stress, moving on to ozone and some other air pollutants while at Boyce Thompson Institute.
So Robert, when you started to work at BTI, the other programs that we mentioned, like environmental biology, plant stress program, and plant control program, which work on insect pathologies, were well established, and they were still functioning very well, getting quite big grants for big research projects. But during the 90s, everything was slowly changing because of technological developments in both genetics and then genomics. So I guess this technology also had influence on how molecular biology was integrated at both genetics and genomics level. And slowly these other programs were losing their importance at BTI, and molecular biology was taking over. How do you remember this kind of slow transition towards, I think the program will at some point change to centers, and then gradually, they were dissolved?
Yeah, so I think the dissolution probably happened after I left, but I don't, so I don't remember that so vividly. But yeah, the funding opportunities changed. So, for example, I believe that folks like Dick Staples and others had been funded not only for very excellent fundamental research, but also screening for biocontrol agents, perhaps through industry, I believe through industry support. The environmental biology program had gotten really great support from the Electric Power Research Institute and probably the Environmental Protection Agency and so forth. So the good news, I guess, for example, there is that as scrubbers were added to the smokestacks of these horribly polluting coal power plants, there was less of those air pollutants. That's my recollection, and so that led to diminished funding. And then I think sort of from maybe from the board and the president's office and so forth, the desire to ramp up molecular biology and make it more part of the fabric of BTI became very, very, very apparent.
Yeah, I think also on the environmental front, some standards were established, right, for the levels of pollutants in the air that could be acceptable or not acceptable. And I think environmental program played an important role in helping to establish some of them.
Yes.
But once they were there, this function maybe was less important.
Yes, and we can really hope that within 10 or 20 years, all the concerns about greenhouse gas that we have right now will be replaced by other concerns, in a similar kind of evolutionary way, yeah.
Robert, I would like you also to talk a little bit about the collaborations with various departments at Cornell. So BTI is lucky to be placed on the campus of Cornell University, and I think that builds certain synergies between people working in similar fields. Could you talk about your own experience when it comes?
Sure. So collaborations ranging from teaching, for example, I taught with Ray Wu, who was a really distinguished plant science, well, general molecular biologist and plant molecular biologist. I believe he was in biochemistry. Taught with Andre Jagendorf for a while while Andre was still teaching. But I think most, and Steve Howell, by the way, had I believe, a huge influence on the trajectory and quality of the plant molecular biology teaching at Cornell. My memory is that he really was influential in creating a whole series of like a curriculum series for plant molecular biology from his office at BTI, with a great impact on generations of Cornell students. Then there certainly were strong research collaborations. But my recollection is that David Stern was collaborating with Karen Kindle and Maureen Hansen down Tower Road in the biotech building on campus. Steve was collaborating with Leon Cochin across the street at the USDA lab. So, yeah.
And your own collaboration with the Department of Genetics and Development at the time, which later became Genetics, Genomics and Development.
Right.
Was that mostly research collaboration or teaching?
Well, it was mostly graduate education, I would say. And so, for example, as were the other members of the plant molecular biology program, Steve and David, I got deeply involved in graduate recruitment, in genetics, and I believe also in plant biology, the graduate program in plant biology. Chaired some years the recruitment effort and things like that. So, yeah.
And Robert, could you tell us how was the social life at BTI? There was a big difference between, institute being placed in Yonkers, New York, and it was a huge adjustment when they moved to BTI, to a new building, a new environment. But how do you remember this small community at BTI?
Yeah, mostly I remember it very positively. There were certainly stresses and strains that one experienced. I guess we were the young upstarts or something, and as you said, adjustments. I never actually visited the estate, or so I've never actually been on that land. But clearly that was a different era, a different family kind of environment. But in fact, I felt quite a family environment at BTI as well. So for a while, there were Friday afternoon social activities, very, very informal, down in the lobby, which is now the area with the fishbowl, the area with the walled off, the glassed off area, which wasn't glassed off at the time. People would get together for cold beverages on Friday afternoon. I don't remember if it was once a month or just, and some of us would then go out to dinner together. Again, very informal, very casual, and a lot of fun. And it was not, it was not only the professors. It was actually during that time, my partner Jill and I became very good friends with Larry Willard and his wife Carol. Larry was, I believe he was like the head of the mechanical department at BTI. The folks who kept the building running, and in Larry's case, the growth chambers running. Thank goodness. Without those growth chambers, we couldn't do anything. So postdocs, maybe some graduate students, staff, faculty would go out and maybe have pizza and beer afterward. And it was just a very nice community.
So as we mentioned before, Robert, a lot of changes in technological changes were taking place in the 1990s, both in genetics and genomics. And I think this will allow us to discuss your transition from BTI to private sector. If you could explain why you decided to do so in connection to technological changes.
Sure. I can perhaps illustrate this with a story, which was that I was teaching a graduate, I think in genetics and development, a graduate seminar, and one of the papers that we talked about was the first paper on gene chips. So this was this amazing technology that Patrick Brown, who then more recently invented Impossible Foods. That was his another thing that he's a real polymath. They had figured out how to use photolithography from the computer chip industry to actually synthesize tens of thousands of small pieces of DNA on a small piece of silica, right? And you could then use that to do a lot of things. I think at first to look at, instead of, oh, I've cloned a gene and now I want to see where it's expressed. And does it respond to ozone? Does it respond to ultraviolet light? Does it change in its expression during development? You could do it for 10,000 or 20,000 genes at a time. There's an example of where my appetite was whet by some teaching that I did at Cornell. Well, this sort of these and then sequencing technologies, instead of doing all of the sequencing in your lab on a single gene, would take 3 months of hard labor to do it. All of a sudden, fast and efficient approaches to sequencing became available. And those really, at that moment in 97, 98, were not really available in most universities. And they were becoming more rapidly available in industry. And so when Bill Timberlake approached me and said that they were building this startup on steroids with funding from Monsanto, was I interested in becoming one of the founding directors, science directors. I got very interested in this.
Okay, and what you were able to do as a science director, as a scientist?
At Sirion. Yeah. Well, so we did shotgun sequencing of the Arabidopsis thaliana genome, this plant that I've been studying for now a while, 12 years or 13 years. So in some ways, we were the first people, just to brag a little, to sequence the Arabidopsis genome. Now, we did it using this shotgun method that Eric Lander and others had proposed for sequencing. But really, the reason we did it is the community, funded by government agencies and so forth, the international community was creating this beautiful genome sequence for a different strain of Arabidopsis. And we wanted to be able to do mapping using these polymorphisms, these differences in the DNA across the genome, to be able to find genes that were of agricultural and agronomic importance for Monsanto using Arabidopsis. And so that's an example where we did shotgun sequencing of the Landsberg erecta genome. We're able to compare it back to the reference sequence done in the public domain, this very high quality sequence that we still use. It's a fantastic resource. And somehow, and it was Steve Rounsley and others who really pushed this, we somehow managed to get the Monsanto lawyers to agree to let us actually release the sequence so that the rest of the world could could use it. And George Jander, who's been at BTI now for 20 years, I don't know, I have to count. Was a staff scientist in the department, and he is the first author on the paper that described the shotgun sequencing and the use of the shotgun sequence. So, and then he came to BTI when Sirion ended its short and happy life, short and intense life.
Okay, so for a couple of years in the early 2000, you were able to do this cutting-edge research thanks to technology that was available in the private sector. But the universities, I understand, were catching up. Yes. And gradually established also genetics and genomics departments. Before you started to work at the Michigan State University, you worked briefly at the National Science Foundation. Could you explain what happened?
Sure. So Sirion had 4 years of rocket growth, really amazing growth. And then Monsanto said, okay, time to incorporate into the larger company, and I didn't do that. We didn't. The family didn't want to move, and we wanted to stay in the Boston area for a while. So I took a job at the National Science Foundation, the Plant Genome Research Program, and this was a really great opportunity to learn, both to help select grant proposals for funding, and to figure out and learn what was going on back in academia, mostly in academia and research institutes such as BTI. So it was a really great opportunity for that.
Okay, so that was helpful to you also as a researcher or scientist to see what was happening in the academic institutions. That's right?
And to learn how to write grant proposals even better. I mean, if you read hundreds of grant proposals and you're paying it and paying attention, you can learn a lot about grantspersonship, right?
And I guess until today, the NSF is the main foundation providing huge grant for research, right? I think it might be main federal agency.
Yeah, certainly. Broadly speaking, that's right. For plant and related science, Department of Energy has become quite important over the years. Yes, more broadly important. They've been interested in certain aspects of plant science for a long time. The USDA, of course, the U.S. Department of Agriculture, has spends a lot of money on many things, including research.
So, Robert, since 2005, for almost 20 years now, you have been affiliated with Michigan State University, and you work as a professor in biochemistry and molecular biology. Recently, in 2021, you were named a University Distinguished Professor in many fields related to plant biology. Could you talk a little bit about your academic career, about your work as a scholar and scientist, and what was happening during the last 20 years?
Right. So, yeah, a lot has happened in the last 20 years. But in brief, I went to MSU because of this tremendous research environment, a tremendous community, which is different now than it was then, but is still tremendous. And I guess I was hired in part because of my knowledge of doing genomic style genetics research, big projects, collaborative, multidisciplinary projects, and so forth. So the first proposal that we put in was to NSF, and it was for a very large so-called phenomics project. So where one is phenotyping a large number of plants efficiently and so forth. And our focus was on the chloroplast, which is the defining organelle of photosynthetic organisms, eukaryotic photosynthetic organisms. So this was a multidisciplinary. There's resonance here with, say, what happened at BTI with reaching across and benefiting from other people's expertise and so forth. Looked at maybe mutants for 3,000 different Arabidopsis genes. A lot of fun. Pivoted then back to my interest in the kind of metabolism that only plants, well, that's not quite true, that plants are masters at, which is called traditionally secondary metabolism or plant natural products. We now call it specialized metabolism. All of these amazing compounds that metabolic pathways that plants have that produce nice tasting, nice colored, often pharmacologically active compounds. So I like to think of the fact that we study the poisons that plants have evolved to make to protect themselves from their environment. And there's always this arms race going on between the plant and the thing it's protecting itself from, right? Which leads to, not intentionally, but through evolutionary mechanisms, to diversification of those biosynthetic pathways in the plant. And then the other organisms go, oh, okay, they start evolving protective mechanisms, detoxification, et cetera. And actually Cornell is this campus is a historically and presently a real hub for studying those kinds of interactions. So I've been interested on the biochemistry side, finally got back to studying in the last 15 years how proteins evolve, right? This thing that we just was not really possible to do in 1979 through 80, 86. We now have comparative genomics. We can sequence genomes. These technologies allow us to sequence the transcripts, the mRNAs, the genomes from hundreds, thousands of plants to say, oh, look, that plant makes this compound, that plant makes that compound. They look similar to each other. Are they made similarly? Well, sometimes the answer is yes. Sometimes the answer is no. They were reinvented by evolution, by proteins randomly evolving and so forth. And so that's the kind of stuff that we do now in my lab. The postdocs and until recently graduate students in the lab at MSU.
But it sounds to me, Robert, that these advanced technologies in genetic screening, they are always a core of your research, and they help you to advance and do.
Yeah, and I'll call out one that we haven't talked about quite yet. We've talked about genomics technologies, but phenotyping, being able to measure the sort of sum and substance of an organism. Differences in humans, eye color, hair curliness, skin pigmentation, and so forth, the phenotype, right? So because we're interested in metabolism, these crazy molecules that plants make that are sometimes beneficial, often beneficial to humans, to actually analyze those, we can't use a microscope the way you would if you want to look at pollen or skin cells. Or we started back at Sirion when George and I were colleagues back at Sirion. We started a very productive collaboration with analytical chemists, a woman named Lily Lee and others, again cross-disciplinary, to invent ways of, instead of measuring a few molecules in a small number of samples, to measure large number of molecules in thousands of samples. And then at Michigan State, I've been really fortunate. At about the same time that I was hired, a fellow named Dan Jones was hired to direct the small molecule mass spectrometry core. Mass spectrometry are these amazing tools that used to fit in a whole room and now fit on a bench top. You have them at BTI, as we have them at Michigan State. And these collaborations, these facilities have allowed us, we've probably run, I'm just going to say 500,000 samples through these these advanced phenotyping technologies, and that allows us to do things that I don't think we ever would have. Well, maybe we would have guessed that we'd be doing 30 years ago, but I don't think I was that creative to be able to think or far enough thinking ahead to think that we'd be able to do what we can do right now with advanced analytical chemistry.
In recognition of your research at Michigan State University, you were also, you became a fellow of the American Society of Plant Biologists and also of the American Association for
Advancement of Science.
Advancement of Science, right? And I just wanted to ask you from your perspective as being fellow in this societies or association, could you talk about the most exciting developments in field of studies over the last 20 years? I understand research is one thing, but maybe you see a broader perspective also as being fellow and also leading, I think, at some point, the American Society of Plant Biologists as a director, right?
Right. Well, I think one of the things that doesn't relate directly to science, but is to technology, but is super important for the future of technology, is the instead of tacit recognition, but instead the active recognition of the importance of the workforce, the broad workforce. I know that I've known people who get very annoyed by the idea of the term human capital, but this economic concept, sociological concept that empowering the full workforce, the full population of people, everyone, to participate in this case, biology, biotechnology, and so forth. We think of it as diversity, equity, and inclusion. I think from a very practical perspective, and it's super important to think of it that way, but also just making sure that that our future workforce is as representative of and as diverse as the world's population. And working towards that actively instead of saying, oh, yeah, that's a nice thing. So, for example, at ASPB, American Society of Plant Biologists, there are very strong recognition and activities towards making sure that young scientists of all backgrounds, colors, socioeconomic backgrounds, and so forth, come to our conference, read our journals, are involved in learning how to be a scientist through doing, through participating. So don't know if that's the kind of thing you were thinking of, but that's at least one non-technical trend that's super important. Of course, the internet allows that. It also sometimes influences in ways that aren't all that great.
That's great. That's great. But that's the broader perspective on how science is evolving. What is important in connecting different areas of science and also involving people of different backgrounds and origins, right? In science. So at the end, I would like to ask you about your teaching philosophy and about what you like the most about your work as a scientist and scholar.
Yeah, another small question, right? A huge question, really. So in terms of teaching, I'd say train. If I may just change the word slightly to training, because I've learned to benefit so much from the training that I've received, the multidisciplinary training, the training through working with scientists who've trained in diverse fields, analytical chemistry, computational science, et cetera. I've really I'd like to focus on training the next generation of scientists, policy makers, educators, and the way that I've tried to get involved in this to try to amplify a signal, of course, within the lab, within our research group and our research collaborations, of course, but also then at Michigan State, we developed a research experience for undergraduate programs, research experience for undergraduate program called Plant Genomics at Michigan State University. It's been running, 15 years, something like that, with a bump from COVID, a disruption from COVID, now run very ably by my friend and colleague Cornelius Barry. If you think about 15 times, on average 14, 15 students per year, it's a lot of students who've gotten an opportunity to participate in research. For the past 10 years, I've been the founding program director of the Plant Biotechnology for Health and Sustainability NIH T32 funded training program at MSU. Again, a way of in this case, not undergraduate education, but graduate students, giving them not only funding, but some training that is complementary to the training that they're receiving in their graduate programs, and helping them think about what their next step is. Most students don't want to or choose not to. I'll say it positively. Most students choose to go into non-academic career paths, and one of the things in this biotechnology training program is we give students an opportunity. We give them funding so they can go and spend a summer, a semester at a company, at a government laboratory, and so forth. So learning by doing, really giving people a chance to learn by doing and to broaden.
And again, Robert, this personal question: what do you enjoy the most as a scientist, as a teacher, and in your research in general?
Yeah.
Or maybe somebody who inspired you as a teacher or colleague, and you apply his lessons to your work.
Right. Well, so I'll answer the second part first. I'd say here at BTI, let's just focus, because there have been so many, I could go on for a half an hour. Steve Howell has been extremely influential, and he may not realize it, and hopefully he'll get a chance to see or read the transcript from this and know that it's true if he doesn't. He encouraged me to go to conferences that were not the Arabidopsis meeting, or to branch out and to put myself out there. And so I try to do that with other people as well. Ralph Hardy, it's really interesting, and this may just be my lens of history, but Ralph, he spent his entire career pre-BTI in industry, mostly at DuPont. Despite that, and you might think, oh, very hierarchical, very top-down. Ralph was not a micromanager. I now realize that he hired people and let them do their thing, which was really quite wonderful. Also, Maureen Hansen, not at BTI, but in the environment here at Cornell, she was an inspiration to me as someone who looked beyond her own self-interest and really spent a lot of her time and energy generating programs that were beneficial to a large number of people. So that's the second part. The first part again.
What do you enjoy the most?
Yeah, the success of students. That is the success of people who have been through the laboratory. The groups at BTI, and I'm not going to name names because I will leave people out. But then the people who were and some who still are in the lab at Michigan State, as well as some of the folks who I've interacted with in industry. I mean, George is one of the examples of people at Sirion who was so much fun to work with. So I will name at least one name, BTI related. So I think watching their success is really cool, and watching them actually, in some cases, achieve things which I which make me, if you want, which humble me, which is a nice thing. It's a good kind of humbling.
All right. Thank you so much, Robert.
You're very welcome. Thank you. It's been a pleasure.